Genetics and Genomics in Medicine, Chapter 6, Principles of Gene Regulation and Epigenetics. Overview. In this module, we'll consider processes that regulate our genes and how they're expressed, with an emphasis on transcription, post-transcriptional modification, and translation. The learning objectives for this chapter are to be able to describe basic regulation of gene expression, which is what we considered in our previous module, to relate chromatin modification and epigenetic regulation to modifying gene expression, which is what we're emphasizing on this particular module, and to explain underlying epigenetic abnormalities which underlie common diseases, and that will be in our next module. Module 2, Chromatin modification and epigenetic modulation. Epigenetic mechanisms. What are epigenetic mechanisms? These are all heritable changes that are not the result of an alteration of DNA sequence. So alteration of DNA sequence refers to those things that we talked about in the last module that could affect promoter or enhancer, any kind of cis-acting element because the actual DNA bases have changed. But an epigenetic mechanism refers to things such as DNA modification, so actual modification of the base itself, and histone modification. So again, recall that in chromatin we have the DNA wound around histones to form nucleosomes, and that causes the chromatin to be condensed, in which case it's transcriptionally inactive, or other modifications can cause the chromatin to become more relaxed, and in that case it's accessible by the transcription machinery and therefore becomes transcriptionally active. Another factor that can be thought of when we think of epigenetic mechanisms is alteration of nucleosome positioning. So let's ask the question, what is the underlying effect then of epigenetic changes? So that would be is that it alters the chromatin environment. So again, as I said, the chromatin environment, when we talk about condensed versus non-condensed chromatin, that in turn attenuates transcription and thus affects gene expression. So altering the chromatin environment affects gene expression at the level of transcription. Okay, so that's something to keep in mind. Let's take a look at some examples of epigenetic phenomenon, and they're quite common in things like development and gametogenesis. So, for example, we have epigenetic reprogramming that takes place in the early embryo. And so when we talk about the gametes, the egg and the sperm, their genomes have specific epigenetic marks. That is, that either their DNA is altered in some way, modified in some way, or their nucleosomes, the actual histones are modified, and this actually changes when they combine to give the zygote. So during fertilization, and the gametes then combine, egg and sperm cells combine to give the zygote. Their genomes are reprogrammed to erase the vast majority of these inherited epigenetic marks. But then, by the blastocyst stage of development, the cells of the inner cell mass become are then pluripotent, and that means that they could give rise to any cell. So remember, by having epigenetic marks, you're attenuating gene expression, and that is contrary to pluripotency. But then, as development continues, then epigenetic marks are reestablished in the descendants of the cells of the inner cell mass to establish different cell lineages and permit cell differentiation. So that's one phenomenon that takes place in early development that involves epigenetic reprogramming. There's also epigenetic reprogramming in gametogenesis, and that's where we make the egg and sperm cells. And this is readily detected as a wave of genome-wide demethylation that occurs during germ cell development. And it erases all of the parental epigenetic marks, and then it's followed by de novo DNA methylation to reset the global patterns of DNA methylation and gene expression. Another example of epigenetic phenomena is the establishment of constitutive heterochromatin. So there are some genes that are constitutively not transcribed. They're constitutively silenced. So remember, heterochromatin is transcriptionally inactive. And an example of that is centromere establishment relies on a specific histone H3 variant as CENPA. Okay, and that is transcriptionally inactive. Another example is X chromosome inactivation. So, as we know, males are XY and females are XX. But to make sure that the gene dosage in females is not above that off the X chromosome than it would be in a male. One of the X chromosomes is actually inactivated during development, and that then becomes. We'll talk a little bit more later about that. That's an interesting phenomenon, but that is an example of epigenetic phenomenon. So there are modifications that occur either at the nucleosome level or at the DNA level that silence that extra X chromosome that females have. And then there's genomic imprinting, and that is actually, again, silencing of one allele according to the parent of origin at diverse loci. So oftentimes in terms of gene dosage, you may have gene expression off of only one of the alleles, one of the parental alleles. You won't have expression off of both. And that's important in many cases to attain the proper amount of gene dosage. And when I say gene dosage, that, to simplify that language, that means how much protein is being produced off of a particular gene. And then lastly, we have position effects causing heterochromatization. So these are large-scale changes in DNA that cause genes to be relocated to a heterochromatin environment where they are silenced. And we'll talk a little bit more about each one of these examples later on in this lecture. It's important to consider that changes in chromatin structure actually influence altered gene expression. So in our previous module, we talked about how changes in the DNA sequence, the actual sequence of the DNA or cis-acting elements, can alter gene expression. But changes in the chromatin structure itself can produce altered gene expression. And what does that mean exactly? So for example, if we have a gene that's actually functional, that has a promoter, it's meant to be expressed, if it's actually located in chromatin that's highly condensed, then it wouldn't be accessible to transcription factors, and that gene would be silenced, even though it is intended to be a functional gene. But if the chromatin structure where it's residing is altered and then adopts a more open, relaxed conformation, then the protein factors can bind the promoter and regulatory sequences in order for transcription to be initiated. On the other hand, we can also think about a gene that is normally expressed, and if transposition or translocation or inversion, some other type of gross change occurs to relocate the positioning of that gene, then that gene would be silenced. And that particular example is an example of a positional effect. And so when we think about that, what are the things when we talk about then, simply stated, about more open and relaxed conformations, what are the sorts of things that lead to that? And that's actually chemical modification of the DNA strands and histones. So that would be covalent modification of the DNA strands and histones. And what does that actually look like? Well, examples are at the DNA level, methylation of the cytosines. And that is really the only only possible covalent modification of DNA itself is methylation of cytosines. And then when we talk about heterochromatin, different things that kind of alter nucleosome positioning, and that in turn leads to opening of the chromatin or restricting of the chromatin. Those are post-translational modifications of the histone. So we can actually alter what the histones behave in terms of chromatin structure based on the modifications in terms of acetylation. Those are examples of things that can happen. Methylation, acetylation are different, and sometimes even phosphorylation of the histones can alter the chromatin structure. And then lastly, as we said, the most extreme example of altering the environment in which a functional gene finds itself would be translocation or inversion, where we're actually literally moving the gene itself to another location that might be transcriptionally inactive. So we often hear the term epigenetic marks in the literature, and epigenetic marks refer to patterns of DNA methylation and histone modification. So recall histone modification is a covalent modification of the proteins that comprise the nucleosomes, the histone proteins, and acetylation is one very common example of that modification. So those are referred to as epigenetic marks. And there's actually many different enzymes that are responsible for carrying out these epigenetic marks, and people can categorize them into writers, which are the enzymes that add the modifying groups, and erasers that can remove the modifying groups, because this is a dynamic process. So we have to think about the concept and the notion of chromatin remodeling, and so over time and depending on the needs during development or different physiological states, we can make the chromatin more or less accessible, more or less condensed, and so when there isn't necessarily a permanency to these modifications. So that is dynamic. They are reversible. And then so chromatin remodeling is a very important part. In addition to the modifications of the histones themselves, there is something known as histone substitution. So there are variant versions of different histones. So in addition to the histones themselves becoming modified, there are variant versions of the histones. So for example, histone 2A has a couple different variants, as does histone 3. So that's something interesting that we will see soon.
So in our previous slide, we had talked about modification of histones in nucleosomes, and we talked about the different types of modifications that we see in nature: acetylation, methylation, and phosphorylation. We actually looked at the different types of amino acids and what types of modifications would be common in those. And then we also mentioned that there are specific enzymes that are responsible for these histone modifications. For example, histone acetylase transferases, or HATs, histone deacetylases. And we said that these processes are dynamic, and so we have transferases that are transferring these functional groups, and then enzymes that carry out the reverse function. So now let's discuss a little bit about the effect of modified histones as well as histone variants on chromosome structure. So it's accepted and understood, well known, that histones undergo many types of covalent modifications, as we have described previously. And typically methylation of histones is associated with transcription activation and chromatin relaxation. Whereas the reverse process is associated with the chromatin condensation. And so we'll have a slide that kind of exemplifies that in a bit. But you do have certain types of modification that are associated with overall chromatin relaxation, and then in turn we all know that chromatin relaxation then promotes transcription activation. And then there are several different types of histone variants, and we've talked about that. So in addition to histone modification, core histone proteins can be replaced by minor variants, and these predominantly are of the histone classes 2A and 3. And the variants typically differ from the typical histone by just a few amino acids, and the minor histone variants are synthesized throughout interphase and are often inserted into previously formed chromatin by histone exchange reaction. And a well-studied example of this is the CENP3A, a centromere-specific histone H3 variant that's responsible for assembling kinetochores at the centromeres. And that is just one example. The modified histones and histone variants generally do work together with DNA methylation and demethylation to regulate gene expression. Next, we're looking at a table here from your textbook that talks about histone modifications that are characteristic of different chromatin states, and this just gives you some examples. If we talk about histone 3, and we look at the fourth lysine residue, so the nomenclature H3K4 is describing histone H3. And specifically we're looking at the fourth lysine, and we can see that in that instance we can have 2 methyl groups attached or 3 methyl groups attached at that particular lysine residue, and that's associated with an active promoter state. And then as you go on the table, there are different descriptors. H3K9, if you look at the 9th lysine residue of histone H3, that an acetylation at that 9th lysine is associated with activation of promoters, and that a methylation, methylation attachment of 3 methyl groups at that particular lysine is associated with inactivation. Now, please don't think that you need to memorize this entire table. That's not necessary, but it's just sort of giving you a flavor of the different types of histone modification that are characteristic of different chromatin states. It gives an overview here. You can see there are different places where we're talking about euchromatin and heterochromatin, things that are are at promoters, things are at enhancers, situations that are at within the gene bodies themselves. So there's a whole variety. So it's something that's very widespread that you can see. And then the next table talks about the different histone variants that are present in nature. And remember, those can be exchanged out in the chromatin. So there are a couple of variants that are shown in this table for histone H2A. And they're associated with different types of phenomenon. So the H2AX, for example, is important in DNA repair and recombination. It's actually introduced at sites of double-strand breaks, and so it's what should be the take-home message of this this description about chromatin remodeling is that depending on what processes are taking place, it's very important that chromatin be capable of being compacted, because that's how we're fitting it in the nucleus, but also that for different nucleic acid transactions, and we've been focusing pretty much in this chapter on gene expression, and that would mean specifically transcription, but there are other phenomena as well that require access to the chromatin, and so that could be DNA repair, that could be DNA replication, that could be chromosome segregation during cell division. So there are a variety of processes that necessitate the chromatin becoming decondensed or becoming condensed, as the specific case warrants. And this slide, which is really a very nice summary type slide, it shows the contributions made by DNA and histone modification to different chromatin states in neighboring regions on a chromosome. And it shows all of the different things that we've been talking about, starting from DNA all the way down to the chromatin itself, and giving an overview of the different types of modification and how they really relate to inactive versus active gene expression, that is, in this case, active or inactive transcriptional states. And when you look at the topmost part of this figure, the blue line represents DNA, and the yellow circles represent DNA methylation. And so you can see, as we had stated earlier, that DNA methylation is associated with transcriptionally inactive states, and that hypomethylation, or low levels of methylation are associated with active states. And you remember, down at the bottom of this figure, we have the left side are scenarios that are associated with inactive transcriptional states, whereas on the right-hand side of the figure are the phenomena that are associated with transcriptionally active states of the chromatin. Below the line that depicts methylation, we have places that are showing histone-related modifications, whether that be actual histone modification itself or histone replacement, and on promoting inactivity of transcription. We have on the left, there are different types of modifications, methylations, and shown in large yellow circles that are bound by a red border. So large yellow circles with red borders are indicative of inactivity and of transcription. And so you see that certain forms of methylation of the histones are affiliated with transcriptional inactivity. And here we still show the little yellow circles, and so the DNA that's on these nucleosomes is hypermethylated, and that's consistent with a transcriptionally inactive state. Conversely, on the right side of this particular line are conditions which are characteristic of transcriptionally active chromatin. And you can see that in this case we've had some histone replacement. There are fewer numbers of histones there. That, because the idea is to have the DNA less compacted, less engaged with the nucleosome, so there are fewer histones, and that promotes that. And we see here also that the DNA itself on the nucleosomes is not very methylated. In fact, not at all. And you can see there are some different types of methylation. Again, specifically memorizing which types of methylations are pro-transcriptional and which ones are anti-transcriptional isn't necessary, but it's just to show you that they're different. So we wouldn't think that the same type of methylation would be associated with both activity and inactivity. And then as we further go down the diagram, we notice that there are different types of chromatin effector proteins, some which promote chromatin compacting, and some which promote nucleosome repositioning. And again, repositioning means that we're trying to free up the DNA from the histones, not have it be associated with nucleosomes, and it shows very clearly here why that's the case, because the DNA needs to be accessible to the RNA polymerase and the other general transcription factors so that transcription can take place. Transcription cannot take place on a scenario that is shown as on the left-hand side of this figure, where the DNA is extensively bound and wound up within the nucleosomes, and hence you have a very condensed chromatin. So I think this is a really good slide that really summarizes all of what we've been talking about to this point in this module. Now just a few notes about DNA methylation in mammalian cells, because at the end of the day, this is a course that is hoping to focus on human genetics to a large extent. So what are some key points for us to think about when we talk about DNA methylation in mammalian cells? Well, first and foremost, it's important to keep in mind that methylation is the only, and you notice I have this in capital letters, covalent modification of DNA that affects regulation of gene expression. So that's very, very important to keep in mind. This methylation is essential for mammalian development. So when we talk about development and the things that are going to regulate gene expression during development, being able to have DNA methylation is essential to that process. DNA methylation is linked to gene silencing, and so very characteristic of different stages of mammalian development is the fact that certain genes are turned off at specific times, and that's really critical to normal development. And finally, methylation helps to regulate transcription off of transposons. So we know that there are mobile genetic elements, and it's very important that there is a way that we can make sure that any kind of transcription that takes place on these mobile genetic elements is regulated. And when we talk about methylation, that is what really supports things like genomic imprinting and X chromosome inactivation. And we've talked about that earlier, that those are key things that happen during development. Next, let's talk a little bit about DNA methylation, its mechanisms, heritability, and influences during development. So DNA methylation often occurs on the cytosines that are found in CG palindromes. And remember, palindrome is something that reads the same forwards and backwards. So these are where characteristically we see that DNA methylation occurs. It's interesting that the 5-methyl cytosine base pairs with base pairs with guanosine, but it is more susceptible to spontaneous deamination when it's methylated. And if that happens, then the C becomes a T, and then obviously that's no longer going to base pair with G, and that leads to mutation. So in a way, that's not something that we want to promote evolutionarily, and so if we have to have something that's going to deal with methylation, because if that was left unchecked, then eventually we would get rid of cytosines, because once they became methylated, then they would be more susceptible to deamination, and overall that would lead to a trend of disappearing cytosines. And so in nature, what has happened over time is that there are regions referred to as CPG islands, and these are really regions where we have a lot of C and G, and where C and G methylation is suppressed, and this is typically near the promoters, because if methylation, we have said earlier, is really important to regulating transcription, and we get rid of those cytosines that allows us to regulate the transcription, then we lose our control. And what are the enzymes that take place when we talk on a mechanistic level? What are the enzymes that are involved in methylation? And these are the DNA methyltransferases, and they're known by the acronym DNMT. And that stands for DNA methyltransferases. And these are enzymes that serve to maintain existing methylation patterns, and they're specific DNA methyltransferases for different periods in time and specific purposes. So there are a variety of DNA methyltransferases that functions in different ways. So some maintain stability of methylation, some of them are important in epigenetic reprogramming. So they're associated with different types of or different purposes for methylation. It's interesting to know that methylation is something that happens after DNA replication takes place. So when we have nascent DNA means DNA that's just recently been synthesized, then we have to eventually methylate that DNA. And so the DNA methyltransferases help take care of that for us. And then lastly, it's important to consider that DNA methylation is, again, a dynamic process, and it takes place in early development as well as gametogenesis. So when we're forming those germ cells, that's a process that takes place. Okay, this figure shows the pattern of CPG islands and DNA methylation across a gene. So just a hypothetical idealized human gene, and it considers a gene that has a single CPG island located in the vicinity of the transcriptional start site, which is marked by an arrow, and then 3 exons that are demarked as E. So those are the purple rectangles. So the CPG intensity across the gene is illustrated at the top with open circles indicating unmethylated CPG dinucleotides, and circles filled in yellow representing methylated CPG. And so the open circles are unmethylated, and the closed yellow circles are methylated. If the gene is expressed, the gene body shows quite high methylation levels, but the transcriptional start site and upstream enhancer are free of cytosine methylation, allowing the access for transcription factors. Even when the gene is transcriptionally active, the cytosines remain unmethylated within the CPG islands. So this is a nice pictorial way of representing what we had talked about in the previous slide. This next slide serves to review and re-emphasize the function of DNA methylation in mammalian cells, and recall that methylation is the only covalent modification that we see in mammalian cells. It's associated with gene silencing. So methylation is the way that we would silence a gene, particularly if you were to methylate in the region of the promoter. Specifically, the cis-acting elements, promoters, enhancers, are targeted. And methylation is the basis for X chromosome inactivation. So we talked about females, for example, have 2 X chromosomes, only one of which is transcriptionally active. How does it How do we inactivate that other X chromosome? And that's through DNA methylation. DNA methylation also underlines the phenomenon of DNA imprinting. And again, to recap about methylation and methylation mechanisms, DNA methylation is performed by the DNA methyltransferases, or the DNMT enzymes, and during DNA replication of a methylated DNA molecule, because the mature or the parental DNA strand during DNA replication is methylated, and so the each parental strand retains its pattern of methyl cytosines, and the newly synthesized complementary DNA strand is formed by incorporating unmethylated bases. And so, in the absence of any further DNA methylation, you would end up with a hemimethylated DNA following DNA replication. That means that one of the strands would be unmethylated, one would be methylated. So, it's important that we maintain the original DNA replication pattern. So the DNA methyltransferase DNMT1 is normally present at the DNA replication fork and helps to methylate the nascent or the growing DNA strand. And it methylates only on those CG dinucleotides that are paired with a methylated CG on the opposing parental strand. So here we know that the DNMT1 methyltransferase, and that's probably important to know for your exam, that's the one that's associated with maintaining the methylation pattern following DNA replication, and to make sure that the newly synthesized daughter strand has the same methylation pattern as the parental strand. There are other DNA methyltransferases, the DNMT3A and DNMT3B, and these are de novo methyltransferases that can methylate any suitable CG dinucleotide. And they have important roles in epigenetic reprogramming when epigenetic marks are comprehensively reset across the genome at two major stages, and that happens during early development and gametogenesis. This next slide is a nice summary slide that depicts DNA methylation and demethylation mechanisms in mammalian cells. And so, again, we talk about during DNA replication of a DNA molecule, the methylated CG dinucleotides are present on the parental strand, and these are retained during replication. But in the newly synthesized DNA, the methyltransferase DNMT1 is there to maintain that methylation pattern, to add those methyl groups on the newly formed strand to maintain that. Unmethylated DNA can also be generated by active demethylation processes at certain stages of development. So this is a good summary slide to depict that process of methylation and demethylation. So it's a very dynamic process. And our next slide depicts methylation as it takes place in development. So it's important to think about what happens in early development and during gametogenesis. So major epigenetic reprogramming occurs in the early embryo. Egg cells and notably sperm cells have extensively methylated DNA, but each one has different patterns of methylation. And once the sperm has fertilized the egg and introduced its genome, it begins to undergo active DNA demethylation. After the male and female pronuclei fuse, global demethylation of the zygote begins and continues until the early blastula stage in the pre-implantation embryo. Then a wave of genome remethylation occurs, coincident with initial differentiation steps that give rise to different cell lineages. So that's really important. So kind of when you think about it, it's sort of you're wiping the slate clean, and then as we're ready to start needing to, the cells then at that point when everything is demethylated are totipotent, but then we need to establish at some point very early in development cell lineages that are going to support differentiation of the developing embryo, and consistent with that we have remethylation occurring. In addition, significant epigenetic reprogramming also occurs during gametogenesis, and the premadural germ cells that will give rise ultimately to gametes are heavily methylated. And then as they enter the genital ridge, their genomes are progressively demethylated, erasing the vast majority of epigenetic settings. Therefore, de novo methylation allows epigenetic marks to be reset. Next, it's important to take into consideration that there are other factors at play that help to participate in epigenetic regulation in addition to methylation and modification of nucleosomes. And these are the role of non-coding RNA in epigenetic regulation. So non-coding RNA includes tiny RNAs and long non-coding RNA. And long non-coding RNA is very important in genetic as well as epigenetic regulation. And these are actually examples of cis and trans-acting forms of regulation. And mammalian microRNAs are another factor that's at play, but mammalian microRNAs are mostly focused on post-transcriptional gene silencing. By contrast, mammalian long non-coding RNAs are very important in both genetic and epigenetic regulation. And the long non-coding RNAs can work in epigenetic regulation by recruiting specific chromatin remodeling protein complexes to define chromosomal loci. So it's interesting that these, let's talk a little bit more about how the non-coding RNAs can act as either cis or trans-acting regulators of transcription. So most of the epigenetic roles that are described for long non-coding RNAs involve cis-acting regulation. And in these cases, the nascent transcripts of long non-coding RNA may act to tether chromatin modifying complexes that act on neighboring genes. In some cases, a cis-acting non-coding RNA will silence several genes in a cluster, which sometimes involves making very long antisense transcripts. In the special case of X chromosome inactivation, many hundreds of genes across the X chromosomes are silenced by a non-coding RNA. Some mammalian long non-coding RNAs can work as trans-acting regulators. One example of this is the hot air or the Hox antisense intergenic RNA. Genomic imprinting. So genomic imprinting really refers to the differential expression of maternally and paternally inherited alleles. When we think about human genetics, we're accustomed to the idea that in mammals, diploid cells, both the paternal and maternal alleles are expressed, and this would be referred to as biallelic expression. However, for a significant proportion of our genes, only one of the two alleles is expressed. The other allele is silenced. And this is a situation, a scenario, which is referred to as monoallelic expression. That can occur in a random way so that some cells of an individual express the paternal allele and some express the maternal allele. However, for some genes, a paternal allele is consistently silenced, or a maternal allele is consistently silenced. That is, silencing of one allele occurs according to the parent of origin, and this is known as genomic imprinting. Because the natural monoallelic expression occurs for a significant fraction of our genes, the maternal and paternal genomes are not functionally equivalent in mammals. As a result, mammals, unlike several vertebrate species, cannot naturally produce by parthenogenesis, that is, reproduction without fertilization. A haploid chromosome set duplicates within the oocyte. It is possible to manipulate mammalian eggs to artificially make a diploid embryo with two maternal chromosomes, but embryonic lethality always ensues. The maternal genome cannot by itself support development. So that has implications if one thinks about this of things you see in science fiction movies about someone cloning themselves from a single cell of their own selves. That by definition this is impossible, because then you would be just having one particular genome that corresponds to your gender, and that wouldn't be a fertilization event. Parthenogenesis fails in mammals because a subset of developmentally important genes are expressed only if inherited paternally. A different subset are expressed only if inherited from a maternal egg. Our next table talks about gene silencing as it is achieved through various long non-coding RNAs. And so in this table we can see that there are quite a few examples in nature. We have RNA, several different kinds of RNA that are listed on the leftmost column of the table, and then as we move from left to right, it gives the locus of origin, its mode of action, and which target genes there are. So there are actually some genes that are there to code for these long non-coding RNAs, and we see non-coding meaning that they don't code for a protein product, and what genes they affect. So gene silencing is something that's very important, and here's how it takes place. And this next table describes and displays examples of monoallelic expression. We were just discussing how many of the genes in mammals are expressed on a monoallelic basis, and the table discusses the mechanisms by which these are achieved, and discusses whether they're dependent on the parent of origin or independent of the parent of origin. So we did talk about certain situations where we have a specific dependent on the parent of origin. So if we're talking about genomic imprinting, we said that in some cases that can be parentally specific. So for example, all the genes would be silenced from the father's contribution, but they would be active from the mother's. And there are different mechanisms that govern those types of processes versus those that are independent of parental origin. And our next slide re-emphasizes the fact that just as in other types of modification of transcription through epigenetic mechanisms, imprinting, which is a specific case of epigenetic modification, is dynamic, and it can take place, but it also can be reversed. And so here are some examples of reversal of genetic imprinting. And it's quite interesting because imprints can be reversed between generations. So you can have one generation, and then in the offspring the imprinting disappears, and then it can reappear in the subsequent generation, which is, and here's the mechanism by which this could take place. And this last and final series of slides goes over the notion of X chromosome inactivation. And X chromosome inactivation is very important as in mammals, ploidy is really critical. Typically, monosomy is lethal, and the exception of this is loss of the X chromosome, which can result in Turner syndrome. Um, and we've said this before, that any kind of constitutional change in chromosome number, or aneuploidy, abnormal number of chromosomes, or aneuploidy, is usually lethal or results in significant abnormalities. Chromosome loss is particularly damaging, and monosomy, that would be um, loss of or having only one copy of every gene, not and that means only having one copy, and that's different from silencing a particular gene. But total loss of a chromosome is typically lethal. The only exception to that would be a loss of the X chromosome, but even then, that is referred to as Turner syndrome. So we can shut it off, but it's important that it actually is there. And why is this so? Aneuploidies cause problems related to gene dosage. If you have an abnormal number, whether it's greater or less than you're supposed to have, then your gene dosage is going to be off. Um, and there is a difference, really functionally, in the gene dosage when we talk about the sex chromosomes. So females have two X chromosomes, and males have only one X chromosome and a Y chromosome. So how do we deal with that? Well, the way that we deal with that, so that we can ensure the same gene dosage in both males and females, is through the mechanism of X chromosome inactivation. And early in embryogenesis, our cells somehow count how many X chromosomes they contain, and then they permanently inactivate all except one randomly, or at least it seems randomly, selected X chromosome. And the inactive X chromosomes remain in a highly condensed heterochromatic state throughout the cell cycle, and they can be seen as the bar body on the periphery of the cell nucleus. This slide shows some interesting scenarios that can arise from uniparental diploidy and divergent expression of paternal and maternal genomes. So these are abnormal conditions, and it's kind of interesting to consider them now that we have the knowledge to do so. So on rare occasions, a zygote is formed with a genome that's comprised of paternal DNA only, and that produces something known as an androgenetic embryo. And this usually occurs when a diploid sperm, so a diploid, not a haploid, but a diploid sperm fertilizes a faulty egg that lacks chromosomes, as is shown here. So we have on the left-hand side of this figure, you have an egg, pink circle, that's being fertilized by a diploid sperm. So sperms should be haploid, but in this case they're not. And that leads to the production of an androgenetic embryo. And development then produces an abnormal conception that's referred to as a hydatidiform mole with widespread hyperplasia. So the tissues that are formed are hyperplasia of the trophoblast, but no fetal parts. So this does not result in a viable embryo. This scenario. Another scenario that again doesn't result in a viable embryo is the reverse situation, where the zygotic genome is comprised of maternal DNA only, and it produces a parthenogenic embryo, and that will give rise to an ovarian tetrachoma, which consists of disorganized embryonic tissues without the vital extra embryonic membranes. So neither of these result in a normal fetal development. So you can end up with a hydatidiform mole or an ovarian teratoma. Now let's consider another very interesting phenomenon, a little bit lighter phenomenon, and this is the phenomenon of X chromosome inactivation and the calico cat. So, a randomly chosen X chromosome, either in the maternal or X or paternal X, is inactivated on each cell of the 46XX embryo. Now, once this choice is made, it is faithfully transmitted through all subsequent rounds of mitosis. Note that in the oogenia of a female, both X chromosomes are active. Each has an equal chance of being passed on through the egg. Now, in the case of the calico, which is a tortoiseshell and white cat, they're heterozygous at an X-linked coat color locus. So they're heterozygous at an X-linked coat color locus. So one allele specifies a black coat color and the other orange, and then the different color patches reflect clones within the organism in which different X chromosomes are inactivated. And the white patches are the result of an unrelated coat color gene. So in some places you're getting the orange, in some places you're getting the black, and it just depends on what lineage those are derived from, and whether a particular locus was shut off or the other one was. So this is kind of, I think, a very interesting phenomenon that we probably are familiar with from previous biology classes. And on our final slide in this module, we're going to bring up the epigenome. And the epigenome is really the total collection of epigenetic settings across a whole genome. And it's a property of individual cell types and is both variable and flexible because, as we said, that's dynamic and changes with development and over different times. Epigenetic marks such as cytosine methylation and histone modification patterns are not always associated with changes in transcriptional activity, and they are also not as stable as once thought. So we are now appreciating that they are dynamic. Our knowledge of the mechanics of epigenetic regulation remains somewhat limited, and there has been a consortium established, the International Human Epigenetic Consortium, that was established and was launched in 2010 to begin obtaining genome-wide data on the epigenetic settings in human cells. And the initial goal has been to characterize 1,000 epigenomes in 10 years from different human tissues and individuals, with prospectus for translating discoveries into improved human health. So this really is a very interesting thing, but the project has been very controversial because of the variability and the dynamic nature of epigenetics. And this concludes Module 2. Here is a picture that depicts different types of non-coding RNA in epigenetic regulation. In A through C in this picture, we see sequence-specific recognition models in which regulatory RNA base pairs with target sequences. So in A, we see in nascent transcripts. In B, we see one strand of open DNA double helix. In C, we see a closed double-stranded helix forming a triple helix. And then in D and E, we see recruitment of a long non-coding RNA by recognizing a target DNA sequence via complex surface-mediated interactions. And ultimately, this results in local action and co-transcriptional effects.